myself Dr. P. Sakthiselvan, and I'm handling animal biotechnology. And we have discussed about last 5 minutes, and this is the another topic in the fourth unit, which helps you to understand more about in vitro fertilization. So in all the 5 units we have discussed, in the first unit we have discussed about introduction to animal cell culture, animal biotechnology, that is tissue culture, organ culture, and cell culture as a variable product. And in case of second unit we will discuss about the blotting techniques, and in case of third unit monoclonal antibodies, and in applications in diagnosis and also in therapy. In fourth unit we have studied about the artificial insemination, and the another important topic is micromanipulation, which these two topics constitutes fourth unit. And another topic we have discussed in fifth unit, that is transgenic animals, that is we have discussed in last class. So today we are going to discuss about the micromanipulation. So what is micromanipulation? It is refers to the procedures which takes place under the microscope level. So that is the reason it is called as micromanipulation. So it is to manipulate oocytes, eggs, sperms and embryos. That is micromanipulation. So these micromanipulation techniques or microscopic techniques are designed to increase the chance of successful fertilization, okay, in IVF. So that is first there are three methods. One is pre-implementation genetic diagnosis, that is PGD, and ICSI, intracytoplasmic sperm injection, and assisted hatching method. So these are three methods when we are talking about micromanipulation. So we will see what is micromanipulation. So micromanipulation, why it is needed, that is main important question. So why, in the sense, see, when you are talking about micromanipulation, so before we are going to talk about micromanipulation, we should know what is IVF, that is in vitro fertilization. So that is the advanced technique of IVF is micromanipulation, because in case of IVF we are developing a test tube baby with the help of sperm and egg. So sometimes it is not fertilized to produce infant. So such or embryo, such drawback or such kind of negatives can be overcome by the micromanipulation, since it is happen in the microscope level, and it is used with the help of the instrument called micromanipulator. Okay, so we'll see here. During fertilization inside the human body, normally every few sperm, that is when you are physical contact, almost not all the sperms will enter into that ovum. Okay, but in case of micromanipulation, we will inoculate or ejaculate the inoculate, that is a proper name for micromanipulation. We can insert n number of sperms at a time into the egg to fertilize. That is the advantage. And for IVF, during conventional in vitro fertilization, egg from the woman are collected and placed in a petri dish or test tube which containing sperm cells. Okay, so the gametes are then left in an incubator for 7 to 10 days, so for the development of embryo. Okay, so widely used techniques for manipulation is, that is ICSI, intracytoplasmic sperm injection, pre-implementation genetic diagnosis, and assisted hatching. So compared to these three methods, that is PGD and assisted hatching has a major role, since we discussed in the upcoming slides. Okay, so that is pre-implantation genetic diagnosis. What is it? So it is nothing but it is a analysis of single cell which is developed from 6 to 8 cell embryo, okay, in conjugation with IVF, okay, in order to improve the pregnancy. Okay, why consider that PGD, that is pre-implantation genetic diagnosis, in addition to IVF? That is the reason is recurrent miscarriages, because avoid abortions, okay, and avoid genetical diseases and family inherited diseases, and maternal age above 38, and prior failure with IVF, so and balancing for sex. So these are the important parameters or important factors to be considered for the micro manipulation. Okay, so the methods for that PGD is we will discuss here. So first one, we have to isolate a single cell from 6 to 8 developed single cells, and using a flat glass, that is fine glass needle, by puncturing in the zona pellucida. What do you mean zona pellucida? Zona pellucida, it is the communication between follicle cells and the sperm cells. Okay, so that is which helps to fertilization, which helps in the fertilization. Okay, so in skilled hands, this generally does not harm the developing embryo. Each cell is called as a blastomere. That is one. Next one, see that if you see this slide, you will understand that what is PGD. See, blastomere is removed from developed is 8 to 6 to 8 cells. Okay, see that in that they are using a fine glass needle to collect the embryo without disturbing the embryo other cells. That is very important. Okay, so because when you are talking about micro manipulation, that it should not disturb the embryos. If you are disturbed that, then fertilization won't occur in a successful manner. So next one, prepare a metaphase and spread of chromosomes to access the karyotype. Karyotype is nothing but it is to study of chromosomes and number. Okay, so number and integrity of each chromosomes is called karyotype. Okay, so first we have to isolate or analyze a single cell which is developed from the 6 to 8 cell embryo, and prepare the metaphase and karyotype as analysis. These are the very important. And next stage is assessment. There are two types of assessment techniques. One is chromosome painting, another one is PCR. So we will see what is chromosome painting. That is, it is also called as FISH. FISH is nothing but fluorescence in situ hybridization. Okay, so it is nothing but we are hybridizing the cells. Okay, now all the colors, all the cells are in the same color except the fluorescent cells. So because it is probed with the color. Okay, so it is used for identifying the aneuploids. Aneuploids in the sense it is different or incorrect chromosomes. Okay, normally we have 23. So if it is incorrect in the number, then aneuploid. That is called aneuploid. To identify the aneuploid, that chromosome painting or FISH helps you to identify the aneuploid stage or aneuploid factor. Next one, it is limited number of chromosomes can be checked simultaneously because some abnormalities cannot be detectable. That is the another reason. Okay, so next one, this is a pictorial representation for identifying the chromosome painting with the help of FISH. FISH is nothing but fluorescence in situ hybridization. Okay, see that when you see this slide, you will understand that two cells which are in red color, which is painted with red to analyze the abnormal cells. Okay, so that is the reason. See, chromosome 1 in a normal cell painted with a red fluorophore. See, next this is another slide which shows you the multiple probes for identifying the aneuploids. Okay, this is a slide explaining about the screening of aneuploids with multiple probes. Okay, so next one, PCR. What is PCR? So everyone, we know as a biotechnologist, we should know what is PCR because it is important instrument or important method for amplifying the DNA. Because suppose we are having a DNA, suppose 2 strands. Okay, that is we need almost n number of DNA. So that time we need PCR, because it is used to amplify the DNA. It is normally 32 sites has to be performed for the replications. Okay, see that amplification of DNA specific to the gene of interest. Suppose we are having a particular strands, that is 3 dash, that is 3 to 5 dash or 5 to 3 dash, and we have to apply for the primer. See this figure, we are having a target DNA sequence, so we have to multiply for n numbers. Suppose maybe we need 80 or 90 DNA copies, so that time we will go for PCR. So the cycles, which depends on the number of chromosomes or number of DNAs needed. Okay, see that target DNA sequence we are having, that is one strand, that is two strands, that is double stranded DNA. So one is 5 dash to 3 days and another one is 3 dash to 5 days. So we are splitting that into different strands and we are applying the primer. Applying primer is opposite to the nucleotides present. See, for example, in case of primer 5 dash to 3 dash, the primer that stands which ends with TGA, then we have to give a primer as TCA. Okay, that is the reason. See, this is similarly for 3 dash to 5 dash primers, and we have to develop a new DNA and it has to be multiplied in the cycle 2, cycle 3, it goes on multiplying. That is PCR. Okay, so next one. Examples of genetic disorders which are detectable in PCR. First one, Tay-Sachs, that is autosomal recessive, which can be determined with the accuracy of 98%. That is another factor. Next one, cystic fibrosis, which can be detected at the accuracy of 85%. Similarly, Huntington diseases, thalassemia, Duchenne's muscular dystrophy, and spinal muscular atrophy. These are, as more genetic tests are developed as a diagnostic tools, more will be used for the predictive purposes in PGD, that is pre-implementation genetic diagnosis. Okay, next one, what is the limitation of PCR? So PCR, because both alleles may not amplified equally, that is the one drawback, because we are having 5 dash and 3 dash, applying a specific primers for both the strands, and it will go for multiplying with n number of cycles, normally 32 cycles. So sometimes the alleles will not be amplified equally, that is the one drawback. Another one, it does not detect any disorders at a target loci, that is loci of chromosomes. Okay, another mutation may exist elsewhere. Sometimes if you are targeting a particular region, and sometimes some mutations will be takes place in some other places. Okay, to accommodate these limitations, prenatal amniocentesis or chronic villus sampling is usually recommended for supplement as PGD. Okay, so to overcome this issue, prenatal amniocentesis or chronic villus sampling is usually recommended for the PGD. Okay, substitute for the PGD. So next one, that is assisted embryo hatching. What do you mean by assisted embryo hatching? See, we are hatching in the sense we know that we have to catch the particular cells or embryo, whatever, that is catch. So what do you mean hatching? So that we will discuss here. Assisted hatching is an IVF technique in which zona treated prior to the transfer into the embryo in order to weaken the cells. Because see, first we have to weaken the zona pellucida layer. Since it has to weaken, if it is thicker, then likelihood successful is very impossible. Okay, so to achieve the successful transplantation, so we have to weaken the zona pellucida layer. Okay, so next one, just prior to the embryo transfer, developing embryo must hatch out in the outer shell, that is zona pellucida, that is we already discussed. And some embryos still have a thicker shell, when compared may decrease the ability of implant, that is I already told you. In such cases, advanced IVF with assisted hatching is performed, that is the reason. See, suppose if outer layer is in very thicker in color, the thicker, so that time the implantation is very difficult to succeed. So to overcome the issue, so we are go for micro manipulation. It is one of the method in micro manipulation is assisted hatching. Okay. Next one, techniques used in assisted hatching, that is mechanical hatching and chemical hatching, laser hatching. See, we will see there are three methods in the hatching techniques. So we will see what is mechanical hatching, what is chemical hatching, and what is laser hatching. So we will see one by one. So in case of mechanical hatching, here that embryo covers is slit open mechanically with the help of thin long drawn outer glass needle. See this diagram. See, when you see this diagram, the partial sonar dissection has been identified with the help of small thin glass needle. So it will be used to take the cells or take the eggs from the what you call uterus, so to grow in vitro fertilization. Okay, so this is done with the help of machine called micromanipulator. The procedure is, first we have to pierce the zona pellucida with the help of thin glass needle with the both sides, the needle tip being controlled in the perivitline space by eye. That is very important, because we are using our human eye to take the cells, so it should be safely, and it should be in sterile condition, and it is precautions should be taken, so to avoid the disruptions. Okay, next one, the suction of the holding pipette is stopped. So because we are using like you have to suck, because the cells in the uterus, it has to come into the tubes or the needle, so that can be avoided after suction. Okay, so this is the presentation or slide which helps you to understand that. See that we are using a test tube. Okay, we are collecting the cells with the help of needle. Next one, chemical hatching. Chemical hatching is creating a hole in zona pellucida of an embryo by using acidic thyroid solution. Because in mechanical method we are using a glass needle, here we are using a chemical to hatch the cells. Okay, here the solution is acidic thyroids. The procedure for chemical hatching, the fine micropipette containing acidic thyroids solution is brought very close to the zona pellucida region, and the acidic solution expelled gently over a small area with the 20 to 30 mm. Okay, that is the distance, until the zona is dissolved into the inside, because we are only targeting the zona pellucida. So when you are targeting the zona pellucida, you have to take the micropipette with acidic thyroid solutions, and we have to gently add into the zona pellucida, and it easily come into the pipette. After the procedure, the embryos are thoroughly washed in a fresh medium and cultured in the time of transfer. That is very important. Next one, laser assisted hatching. This technique involves the creation of precise gap in the zona pellucida of selected embryos using a 1.48 micro-infrared diode laser. See, first one we are using a needle, second one we are using a chemical, that is acidic thyroids. First one is glass needle, and last one we are using a laser. So laser, here we are using the length of 1.48 micron infrared diode array laser is used to detect or to create a gap in the zona pellucida. So here we are not contact laser or non-contact laser which are used. For both we can use. In case of contact laser radiation has to be delivered directly to the zona, that is called contact laser. See, there are two things. One is contact laser and non-contact laser. In case of contact laser, it has a direct contact with the zona pellucida. But in case of non-contact laser, the laser gamete manipulation as described, which does not have a contact with the zona. It is done using various wavelengths. So procedure is, it allows direct delivery of laser beam through the objectives. Next one, 1.48 micron diode laser is used because it is advantageous overall all over the techniques, and radiation is mutagenic, and the radiation is administered in the contact mode without embryo manipulations. Okay, And next one, assisted hatching done by user light, which is this picture, which helps you to identify, understand easily. See that the embryo is held with a gentle suction using a glass pipette. Okay, so we have the region. I think you might have seen, if you seen, and that you can see that you can easily suck the embryo cells, which is easily with the help of laser or needle. Okay, so the red pilot light provides a visual target for the laser. That is the region we have to succeed. So the white circle marks the safety region based on the temperature. The laser is usually fired 3 times to produce a small hole. That is the reason. See, when you are using a laser, we have to do only 3 times. If you are doing more than that, then the zona pellucida will rupture. Okay, so it destroyed. So then no use of doing micro manipulation. Okay, so and next one, importance of laser assisted hatching. This is specifically designed laser system, includes with the laser which serve as an energy source to create an opening of gap in the zona pellucida. A computer which allows the operation to precisely control the laser energy output, large laser pulse duration and gap size, takes less time and does not expose the embryos to potentially adverse chemicals, because this procedure appears to be quick, precise and chemical free. That is the reason. When you are talking about chemicals, that is acidic thyroids we are using. So that affects embryos inside the uterus. So that creates distractions, or it is potentially adverse to our human body or animals. Okay, so to overcome this is a commonly used advanced technique for the micro manipulation method. Next one, the conclusion here, what I am conclude, micro manipulation simply describes a set of tools and techniques. However, the tools and techniques have played a key role in advancing knowledge of IVF, that is reproductive biology, and expanding the repertoire of clinical methodology and options. They have essentially solved the dilemma of male factor infertility, that is very important, and they allow for the diagnosis and circumvention of inherited genetic conditions, and they hold great promise for further advancement in future. So from here I am conclude that, so when you are using IVF, so that sometimes it fails to fertilize. To overcome this issue, so we are using micro manipulation. It is done by 3 methods. One is pre-implementation genetic diagnosis, and ICSA, that is intracytoplasmic sperm injection. We are directly injecting that to the cytoplasmic region. And another one is assisted hatching. In case of PGD, we have to access 3 types. First, we have to analyze the cell and prepare a metaphase and analyze the karyotype features, and we have to analyze 2 assessments has to be performed. Then is chromosome plating and PCR, and last one is assisted hatching. So assisted hatching, there are 3 methods: mechanical, chemical and laser. Out of which 3, the laser is more important and advantageous method when compared to all other 2. Okay, so from this presentation I am conclude that I have covered almost all the 5 units about the animal biotechnology, and thank you for listening my presentation, and I hope I covered all the 5 units and almost all the topics, because our transgenic animals, artificial insemination, micro manipulation, monoclonal antibodies, these are more important topics in case of animal biotechnology. Thank you for listening my presentation. Thank you.